Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome to the deep dive. Today, we're tackling something pretty counterintuitive in cell biology. Imagine a cell, maybe a cancer cell, getting absolutely crushed, squeezed down. What happens?
Well, you'd think it would just burst, right?
Mhm.
But often it doesn't, especially when cells face extreme physical pressure. Like during cancer spread, they have this instant high-energy defense mechanism.
That's exactly what we're diving into. There's a recent study that's really unpacked this rapid response. It's a kind of mechano-metabolic adaptation. The cell's power generators, the mitochondria, become these emergency first responders.
That's the core of it. The discovery is this mitochondria-derived nuclear ATP surge.
Yeah.
ATP, is the cell's energy money. Suddenly, the nucleus, the control center, needs a huge immediate power boost.
How does it get it?
Through these new structures formed under pressure, they call them Nucleus-Associated Mitochondria, or NAMs. They physically link up.
Yeah.
It's frankly changing how we think about cell survival, especially in cancer.
Okay, let's get straight into the fight. Why does a cell even need this kind of emergency power?
Right, so section 1, the mechanical gauntlet.
When we say mechanical stress or physical constraint, what does that actually look like for a cell? Where does this happen?
It happens a lot, actually. Think about cancer spreading metastasis. For a cell to leave a tumor, it has to, invade the surrounding tissue, squeeze through dense stuff.
It'd be breaking out of jail.
Kind of, yeah. Then there's intravasation, getting into a blood vessel. These vessels have tiny pores, and the cell has to deform massively, contort itself to slip through, often under pretty intense pressure.
And then getting out again on the other side, extravasation.
Exactly. Same problem in reverse. Squeezing out of the vessel into a new tissue, both incredibly physically demanding. The nucleus itself, which is big and stiff, has to get pushed through gaps way smaller than its normal size.
But it's not just cancer cells facing this, right? This sounds fundamental.
Oh, absolutely. It's a universal challenge. During embryonic development, for instance, gastrulation involves massive cell movements, cells pushing past each other into tight spots.
Like building with biological Lego bricks under pressure, sort of. And even your immune system. Think about a T cell hunting down an infection. It has to squeeze through the dense mesh of lymph nodes or inflamed tissue. Any cell needing to navigate crowded environments faces this.
So, the cell gets squeezed. What are the actual consequences? What breaks?
Well, the damage is pretty severe. First, the cytoskeleton, the cell's internal frame, gets warped and stressed. That stress travels right to the nucleus.
And the nucleus isn't designed to be squished.
Not like this. The nuclear envelope, the membrane around the DNA, gets distorted. It loses its shape. And when the container gets deformed, the contents suffer.
The DNA itself.
Precisely. The DNA is packed inside as chromatin. That tight packing gets disrupted, which can lead to actual DNA strand breaks, snapping the code. And this causes major problems if the cell tries to divide later mitotic defects. The literature is clear. Physical stress is bad news for the genome.
Which brings us back to the puzzle. If it causes DNA breaks and screws up division, how do cells, especially aggressive cancer cells, keep going? Why don't they just die off? That's the core question this study tackles. The fact that so many survive suggests they aren't just passively taking the hit. They must have an incredibly powerful, active, and immediate defense, this mechano-metabolic adaptation we mentioned.
Okay, let's unpack the study itself. Section 2. Discovering NMs and the ATP surge. How did the researchers actually simulate this crushing force in the lab?
They needed precise control. Researchers at the Center for Genomic Regulation used these specialized setups, either something called a dynamic confiner or simpler agarose pads. These let them apply a very specific acute pressure to cells.
What kind of cells?
They use common lab lines like HeLa and U2OS, but also, importantly, aggressive breast cancer cell lines. And they compress them down to a height of just 3 micrometers.
3 micrometers? How small is that, really?
It's incredibly small, like maybe a 30th the diameter of a human hair. You're essentially taking a cell that's normally, say, 20 micrometers across and flattening it into a pancake less than a fifth of its usual height. The pressure is immense.
So what was the first clue that something interesting was happening internally?
They started with proteomics. They basically broke the cells apart after squeezing them, separating the nucleus, the mitochondria, the cytoplasm, and took an inventory of the proteins in each part. Subcellular fractionation.
And what did the inventory show?
A really striking result. In the squeezed cells, the nuclear fraction was suddenly full of mitochondrial proteins. Normally, those proteins stay out in the cytoplasm with mitochondria.
So the power plants were suddenly hanging out with the control center.
Exactly, or at least their components are found there. To check if this was real and not just an artifact of breaking the cells apart, they used live cell microscopy.
Watching it happen in real time.
Yes, and it converted the proteomics. Mitochondria literally rushed towards the nucleus, accumulating right at its edge, often within just seconds of applying the pressure. It was incredibly fast.
Which leads us to these NMAMS nucleus-associated mitochondria. What do they actually look like?
It's more than just being nearby. The mitochondria push so hard against the nucleus that the nucleus actually dimples inward. It creates these indentations. The researchers even classified the shapes. Some look like donuts, where mitochondria ringed an indentation. Others were beans, where they clustered on one side.
So the mitochondria are nestled into these nuclear dimples.
Right, tightly packed within them.
Mm-hmm.
But, and this is crucial, they don't cross the nuclear membrane. They stay on the outside, just maximizing their contact surface with the nucleus through this physical deformation.
And how common was this? Did every cell do it?
Pretty much. They found this NAM phenomenon in 84% of the confined HeLa cells. It seems to be a standard operating procedure under this kind of duress.
Okay, so the cell rapidly rebuilds its internal architecture, putting the power plants right next door to the nucleus. Why? What's the functional point? Does it actually change the energy situation inside the nucleus?
That's the million-dollar question they answered. And yes, it absolutely does. This is the core metabolic finding. They used a very clever tool, a periphery sensor. Firster resonance energy transfer. It's basically a fluorescent probe designed to light up specifically in the presence of ATP, and they targeted it inside the nucleus.
So not measuring general cell energy, but precisely the ATP level in the command center.
Exactly. Laser focused. And the results were, well, stunning. The moment they applied the pressure, the nuclear ATP signal shot up, a significant increase, about 60% within just 3 seconds.
60% in 3 seconds. That's like flipping an emergency power switch.
It really is. It's the nuclear ATP surge, and it confirms the whole point of the NAM structure. That extreme physical proximity allows for this incredibly rapid and efficient transfer of energy.
Where is the ATP coming from? Is it a new source?
No, it's generated by the mitochondria right there, using their normal process oxidative phosphorylation. It's just that the energy produced is being immediately channeled, prioritized to the nucleus because of that tight physical coupling facilitated by the NAMs. The architecture enables the surge.
Incredible. So the cell physically remodels itself to deliver emergency power. Section 3. How does it even build these NAM structures so fast? What's the machinery involved?
It requires a coordinated effort using the cell's internal toolkit. The main player seems to be the actin cytoskeleton, specifically what's called de novo actin polymerization.
Meaning the cell builds new actin filaments on the spot.
Exactly. Freshly made dynamic cables built specifically for this job, likely forming a scaffold to pull the mitochondria in and hold them against the nucleus, causing those dimples.
How did they prove actin was essential?
They used drugs. They treated cells with latrunculin A, which basically makes actin filaments fall apart very quickly. When they did that and then squeezed the cells, drastically reduced NM formation.
Yeah.
And the nucleus didn't get those characteristic dimples either. Blocking the actin scaffold effectively disabled the whole structural response.
Did they figure out what kind of actin structures were being built, like ropes or nets?
They did look into that. It seems to be mainly actin elongation, building long, straight filaments that matters. They used another inhibitor, SMETH2, which blocks that process, and it also reduced NAMs. Interestingly, blocking actin branching, which cells use for other things like pushing membranes, didn't seem to affect NAM formation much.
So it's more like rigid support beams or cables being built rather than a diffuse mesh.
That seems to be the idea, yes. Structures to actively pull and hold things in place.
What about other cellular structures? Is anything else involved in holding these mitochondria there? Maybe the endoplasmic reticulum, the ER? It's often connected to both the nucleus and mitochondria.
Good point. The ER plays a crucial role too, particularly in maintaining the association. The ER forms this complex network throughout the cell, like a mesh, and it naturally tethers to both the nuclear membrane and mitochondria.
So it acts like a pre-existing net?
Sort of. What happens under confinement is that the ER network right around the nucleus seems to become immobilized. It effectively traps the mitochondria that have been pulled in by the actin, keeping them pinned there against the nucleus. It's an actin-dependent trapping mechanism.
So actin does the rapid pulling and deforming, and the ER mesh helps stabilize and retain the mitochondria in that position.
That's the picture emerging. It's a multi-component architectural response happening incredibly quickly.
Does the state of the mitochondria themselves matter? We know they can fuse together into networks or break apart into fragments. Does that affect this response?
It definitely does. Mitochondrial dynamics, as it's called, are key. They tested this specifically. They found that when mitochondria were in a more fused state forming connected elongated networks, the NMM formation was more robust.
And the energy surge?
Also stronger. A bolder nuclear ATP surge. They showed this using cells engineered to favor fusion, like knocking out the FIS1 protein, which promotes fragmentation.
Why would fused mitochondria be better for this?
Generally, fused mitochondrial networks are considered more efficient at energy production, especially oxidative phosphorylation. Think of it like a well-connected power grid versus isolated generators. They can share resources and sustain higher output.
And the opposite, fragmented mitochondria.
When they promoted fragmentation, for example, by knocking out MFN1, a fusion protein, they saw lower levels of NAM accumulation and a significantly blunted ATP surge. So yes, the health and connectivity of the mitochondria directly impact how effectively the cell can mount this emergency energy defense.
Okay, section 4. Functional consequences. The cell squeezes, builds NAMs, gets this huge ATP surge into the nucleus. What is all that ATP actually doing in there to protect the genome?
It's not just raw power for keeping the lights on. The key use seems to be for enzymes called ATP-dependent chromatin remodelers. These are the molecular machines the cell uses to open up and access its own DNA.
Wait, but wouldn't the cell want to lock down the DNA tighter when it's being physically crushed? Why open it up?
That's the interesting part. High ATP levels fuel these remodelers to increase chromatin accessibility, loosening the DNA packing. This might do two things. One, some evidence suggests it makes the nucleus itself softer, more deformable, acting like a shock absorber to protect the DNA from shattering under physical force.
Okay, a mechanical benefit.
But perhaps more importantly, when the crushing force does cause DNA breaks, which it likely does, the repair machinery needs to physically get to the damaged site. If the chromatin is locked down tight, the repair crews can't reach the break. ATP provides the energy to open up the chromatin around the damaged site, allowing access for repair.
How did they link the ATP surge directly to this chromatin accessibility?
They use that inhibitor again, oligomycin A, which stops mitochondria from making ATP. They squeeze the cells, but block the ATP surge. And they measured chromatin state.
What did they find?
In the absence of the surge, chromatin accessibility plummeted. Specifically, they saw a big increase in markers of tightly packed, inaccessible DNA, particularly a mark called H3K27E3, which signifies facultative heterochromatin. The DNA essentially clamps down, especially on smaller chromosomes that are usually packed with active genes.
So without the energy boost, the genome becomes inaccessible. What does that mean for fixing the DNA damage that the squeezing causes? You mentioned repair markers like 53BP1 foci.
Right. Confinement definitely causes DNA damage. You see those markers increase. But here's the critical finding. When they block the ATP surge using oligomycin A, or by disrupting the NM scaffold with latrunculin A, the formation of those repair foci was suppressed.
Meaning the damage wasn't happening?
No, the damage was still there. The squeezing still broke the DNA.
Yeah.
But the repair foci, these are actual physical hubs where repair proteins gather, couldn't form properly. The cell couldn't initiate the repair process.
Why not?
Because the chromatin was too condensed and inaccessible due to the lack of ATP for the remodelers. It's like, imagine a bomb goes off inside a secure vault. The DNA damage happens. The repair team, repair proteins, is outside, ready to go. But the electronic lock on the vault door, chromatin, needs an immediate power surge, the nuclear ATP, to open. Without that power, the team can't get in, even though they know there's damage inside.
That makes sense. The surge is the key to unlocking the repair sites, so the damage accumulates unrepaired. What's the ultimate fate of these cells? Does this affect their ability to divide and survive long-term?
It has a massive impact on long-term fitness. They found that just the short period of confinement itself, if the cell could mount the ATP surge defense, didn't really stop the cells from proliferating over the next day and a half. They recovered.
But if you squeeze them, A and D block this.
Then proliferation almost completely stopped. The cells essentially arrested. They traced the problem to a specific stage, a significant delay in S phase progression. That's the phase where DNA is replicated.
So the unrepaired damage prevents them from copying their DNA correctly.
Exactly. It triggers cell cycle checkpoints and stops division. The cell can survive the initial physical insult if its energy defense works, but it cannot survive the insult without that defense. It cripples its ability to proliferate.
This mechanism seems incredibly important, especially for cancer. Section 5. Implications and relevance. Is this NAM structure and ATP surge seen in actual human cancers, or is it just a lab phenomenon?
No, it's absolutely relevant physiologically. The researchers looked at tissue samples, microarrays from breast cancer patients, 17 of them. They specifically stained for mitochondria and looked for those characteristic NAM structures, the nuclear dimpling.
And they found them?
They did. NAMs are present in the patient tumors. But what was really telling was where they found them most often.
Where was that?
They compared the dense center of the tumor core, with the outer edge, the invasive front, where cells are actively breaking away and moving into surrounding tissue.
Where the mechanical stress is highest.
Precisely. And NAMs were found over 3 times more frequently at that invasive front. About 5.4% of cells showed NAMs there, compared to only 1.8% in the tumor core.
That's strong evidence. It suggests this ATP surge mechanism is being actively used by the very cells that are driving metastasis, the ones navigating those physical barriers.
Exactly. It implies that formation isn't just a sign of stress. It might be a key adaptation enabling invasion and survival during that process. It really forces us to see mitochondria not just as general powerhouses, but as these agile, stress-responsive units critical for cancer spread.
And the implications go beyond cancer. The author suggested this might be a universal survival tactic.
Yes, that's the broader potential significance. They propose this is likely a fundamental mechanism used by any cell that needs to survive physical pressure. Immune cells squeezing through tissues, embryonic cells during development. It makes sense that such a critical survival response would be conserved.
So a whole new way cells regulate themselves under duress.
They literally call it a completely new layer of regulation in cell biology, using a localized nuclear energy boost to protect the genome during physically intense times.
Okay, Section 6, limitations and therapeutic opportunities. This sounds like a potential Achilles heel for metastatic cancer, but first, what are the main things the study didn't figure out?
Well, a big unknown is the very first step. How does the cell sense it's being squeezed down to 3 micrometers and immediately trigger the mitochondria to move? That initial mechanosensing signal what molecule or structure feels the pinch and gives the order that pathway is still unclear. That's a key area for future work.
And they were clear this response is specific to the physical act of squeezing, right? Not just any stress.
Absolute crucial point. They tested if other types of stress that cause DNA damage, like chemotherapy drugs, etoposide, or radiation, would also trigger NAM formation in cells that weren't being physically confined. They didn't. No names formed. This rapid structural rearrangement in ATP surge is a highly specific response to physical confinement, not just a general reaction to DNA damage itself. The cell is reacting to the crush, not just the consequences of the crush.
That specificity sounds really important for potential treatments. If only cells under mechanical stress use this, maybe we can target it without hitting healthy, unstressed cells.
That's exactly the therapeutic angle. It represents what they call an underexplored vulnerability in metastatic cells. Because healthy cells in stable tissues aren't constantly activating this AM pathway, you could potentially design drugs that only interfere with this process, hitting the invasive cancer cells much harder.
How might that work? What could you target?
Well, based on the mechanism, there are a couple of obvious strategies. One is to target the physical structure itself. Go after the actin scaffold or the ER network that holds the names together.
Like using something similar to latrunculin A.
Potentially, yes, or developing more specific inhibitors. We saw in the study that disrupting the scaffold prevents the surge and cripples proliferation under confinement. So stop the structure, stop the surge, stop the survival.
What's the other angle?
Target the energy transfer. Find a way to specifically block the delivery of that mitochondrial ATP into the nucleus during confinement. Even if the names form, if the energy can't get through, the protective effect is lost.
So you leave the cell vulnerable to the physical stress, unable to repair the damage.
Exactly. You disarm its emergency defense system right when it needs it most during invasion and metastasis. You effectively leave those repair crews locked outside the vault, crippling the cell's ability to survive the journey.
This has been a really fascinating deep dive. Just to recap the core idea. Under extreme physical pressure, the cell's nucleus sends out an SOS, demanding immediate energy. Mitochondria respond by rushing to the nucleus, forming NAM structures, and delivering a massive localized ATP surge, all to protect the DNA and enable repair.
It's a remarkable piece of cellular engineering born out of necessity. And thinking about the longer-term effects, the study hinted at something even more profound, something you might almost call metabolic memory.
Metabolic memory. Unpack that a bit.
Well, remember how blocking the ATP surge led to reduced chromatin accessibility? They noted that this reduced accessibility seemed to particularly affect genes involved in actin remodeling.
The very process needed to build the scaffolds and allow the cell to move or respond to stress in the future.
Precisely. It suggests a kind of feedback loop. If a cell fails to generate that nuclear ATP surge during a stress event, it not only fails to repair immediate damage, but it also locks down the genetic instructions needed to properly move, change shape, and potentially build defensive structures next time it faces stress.
So the cell's ability to cope with future stress is linked to its energy state during past stress.
It seems plausible. The experience of surviving or failing to survive, that crush could be physically encoded in the chromatin state, influencing future behavior. Which leads to a really provocative final thought. If cancer metastasis relies on successfully navigating these physical stresses time and again, could disrupting the cell's ability to remember or execute this stress response be a novel way to stop its spread?
Targeting the cell's stress history to derail its future?
Yeah.
Powerful idea to consider. Thank you so much for walking us through this.
My pleasure. It's truly exciting work.
That's all for this deep dive. Thanks for joining us, and we'll catch you next time.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.